record it to the cloud.
Great. Okay. So hello, everybody. My name is Evan Shimota. I'm a biology major here at UCR, and I'm in my third year. My PI is Dr. Alan Brailsford, and I'll be presenting on the genetics of social structure and sex ratio in the ant Formica execta. All right. So to begin, we should start with a little bit of background info. In the ant colonies of interest, queen number per ant colony varies. An ant colony can be single queen or multi-queen. Colonies produce female and male sexuals that will eventually reproduce. On the left is a female sexual. Her gaster is shown in the red circle and is fairly large relative to the rest of her body size, and it's round. On the right is a male sexual. His gaster is thinner with a hook on the end. Colonies often specialize in the production of males or females. This leads to biased sex ratios or split sex ratios. Both colony queen number and sex ratios in ant colonies may be determined by supergenes. So this brings forward the question of what is a supergene? A supergene is a region of a chromosome with two or more genes that are inherited together and determine ecological behavior, development, or what an organism may look like. A common example of this are sex chromosomes. In the figure to the right, we have a simple representation in the center to show two versions of a chromosome, with the yellow and orange block representing alternative versions of a supergene that could be inherited. Supergenes have been observed in multiple organisms and includes Batesian mimicry in butterflies, plumage polymorphism in birds, or sex-specific cryptic color morphs in fish, among others. What I am interested in is the potential supergenes in ants. The colony social structure supergene that determines if a colony is single or multi-queen, convergently involved in both Solenopsis and Formica, and this study evaluates species from the Formica genus. So in the Formica genus, a social supergene determines colony queen number. On the right, we have a figure. The x-axis is chromosome number. The y-axis is FST between single queen and multi-queen colonies. FST spans 0 to 1, 0 meaning there is no genetic differentiation, and 1 meaning it's always differentiated or fixed. In the study, Formica execta is the species of interest. As you can see in the figure, there is a region of high density genetic differentiation over chromosome 3, and this represents the potential social supergene. In the related species, Formica glacialis, chromosome version is associated with whether a colony is male or female producing. On the y-axis in the figure, we have chromosome number, or on the x-axis, we have chromosome number, and on the y-axis, we have how associated a particular single nucleotide polymorphism, or SNP, is with sex ratio. Again, we see a cluster of SNPs located on chromosome 3, and this represents the split sex ratio supergene in Formica glacialis. By looking at the FST plot, we can see there are two distinct regions of high genetic differentiation on chromosome 3 in the related species Formica glacialis. This shows that glacialis has a supergene for both social structure and split sex ratio located adjacent to each other on the same chromosome. So this raises some questions about Formica execta, specifically what is controlling colony queen number and what determines sex ratio. So to begin, we should discuss previous research on Formica execta to sort of paint the picture. In the figure, we have female producing colonies and male producing colonies, and on the y-axis, we have relatedness. Female brood in Formica execta are more related to each other than female and male brood in both female producing colonies and male producing colonies. Female producing colonies are typically single queen, and male producing colonies are typically multi-queen. Because of this, relatedness decreases the more queens there are in a colony, and therefore effective queen number influences sex ratio. From this, we can infer that social structure plays an important role in sex ratio. Additionally, other previous research determined that resource availability was found to influence these biased sex ratios. Female producing colonies were consistently located closer to natural food sources than male producing colonies. And when resources per queen were abundant, colonies would typically produce females. And when resources per queen were scarce, colonies would produce males. So this leads us to the two hypotheses in the study. The first one is that Formica execta possesses a single supergene for social structure, and hypothesis 2 is that Formica execta possesses both a supergene for social structure and colony sex ratio. My PI and a graduate student collected 106 Formica execta colonies from Switzerland that contained both workers and sexuals. We found that 19 were male producing, 34 were female producing, and 17 produced both males and females. I performed DNA extractions on 5 sexuals and 5 workers from each colony. And grad student postback and I prepared DNA for RADseq libraries. The UCSD Institute for Genomic Medicine sequenced our libraries, and we received our raw data last week. A grad student has further demultiplexed the raw data, merged read pairs, and removed it after sequence. We still have to align to a reference genome, call SNPs, and create FST and PCA analysis, or principal component analysis on chromosome 3. So here are some of the expectations for what we might find in our research. Here we compare Formica execta multi-queen colonies to single queen colonies, and we would expect the region in the figure with elevated FST to represent the potential social supergene on chromosome 3. In this figure, these are our expectations for the sex ratio supergene. A in the genotype represents a male bias and D represents a female bias. So this FST plot is comparing male bias colonies to female bias colonies. If the findings in Formica execta are similar to those found in Formica glacialis, we would expect that the region here on the figure would be the region of high genetic differentiation on chromosome 3. If Formica execta possesses both of these supergenes, this is what we might expect to see. On the left is the male bias versus female biased FST plot, showing the potential split sex ratio supergene, and on the right is the single queen male bias versus multi-queen male bias colony that shows the location of the potential social structure supergene. This demonstrates the potential for linked supergenes and that male bias and female bias is independent from social form. So based on our hypotheses, we have 2 potential outcomes. If Formica execta just possesses a supergene for social structure, we would expect to see an FST of chromosome 3 that looks similar to the one on the left. That spans the majority of chromosome 3. If Formica execta possesses a supergene for social structure and split sex ratio, we'd expect to see plots of chromosome 3 similar to those on the right. In conclusion, if Formica execta only carries a social supergene, we would accept hypothesis 1. And if Formica execta does have a split sex ratio supergene, we would accept hypothesis 2. I want to shout out and give a huge thank you to Dr. Brailsford and Germán Lagunas Robles for guiding me through my first project, to Germán Lagunas Robles and Zul Alam for helping me prepare RADseq libraries, the Brailsford Lab and the Purcell Lab for allowing me to use their resources to conduct research, and the mini grant review committee members and the undergraduate education for the opportunity to apply for a mini grant, receive partial funding, and experience faculty mentored research. Thank you very much. Are there any questions?
I've got a question.
Yes.
Can I just ask?
Yes.
First of all, awesome talk, really interesting. I was wondering how you think that population structure might influence the plots that you expect.
Is there a slide that I could go to refer?
So let's say, for example, you know, you're collecting from you were talking about how the female colonies, and I'm not an expert in this, so you can forgive me when I make the inevitable mistake. You're saying that the female colonies are more kind of closely related to each other, and I was just wondering whether that close relationship would affect your FST outcomes potentially.
So in our FST plots, we're comparing, in this example, female bias colonies to male bias colonies. So I think it would definitely influence our FST values, because it would help show us where regions of high genetic differentiation between the two populations are.
Okay, so like a strength rather than a weakness, as it is in some other cases where like population structure causes a problem. So then the other question that I had, thank you for that. The other question that I have is, do you think that these supergenes are inversions, or how are they maintained as supergenes?
That is a really great question. I might have to think about that for a little while. I believe they explain the presence of behavior, development, and phenotype, and therefore potentially they're selected for in the environment, and they give some sort of benefit. In this case, we talk about multi-queen and single queen colonies, and how female producing colonies are typically have more resources, and male producing colonies per queen have less resources. So maybe it's some sort of selection to allow for the population to thrive. But if you drop your email in the chat, I can get back to you with a more concise answer.
Thank you, that was great, thanks.
Of course.
Remember any other follow-up questions? All right, well then with no more questions, then I will just open it up for a round of applause to thank Evan for the presentation. Great work. There we go, there's all the emojis. Love it. All right, excellent job.